bored. I want to watch TV. I wonder what's on.
Plasmids.
We're talking about plasmids.
I'm Joelle, the science gal.
And I'm Gear, the genetic engineer.
Plasmids are small circular strands of DNA. They're independent fragments that allow foreign cells to express new genes.
Well, plasmids are able to exist in bacteria. They express foreign genes and synthesize proteins.
Plasmids rule.
No, they rock.
Plasmids are independent of the chromosomes and range from 1,000 to 200,000 base pairs.
Plasmids possess a copy number, which when greater, more proteins can be synthesized. Resistance to antibodies is greater.
They are used as vectors to transfer DNA from one cell to another. This is a plasmid.
Friction endonucleases are used to splice a foreign gene into the plasmid.
Plasmids have been engineered to contain a unique region to be cut, making it linear.
The linear plasmid now has exposed sticky ends, waiting for the proper gene fragment to join the plasmid.
The proper gene is attracted, making the plasmid circular again.
DNA ligase joins the gene fragment and plasmid, making a recombinant DNA.
The new plasmid is introduced into the bacterial cell via transformation.
Here, the plasmid continues to divide, producing many copies of the gene.
We now interrupt this program for some breaking news.
Good news! A new medical breakthrough has been introduced for people with diabetes.
People diagnosed with diabetes have trouble making insulin for their bodies.
In the past, insulin from animals was extracted and inserted into the human body. Although this helped with many cases, some people developed allergic reactions to the animal insulin.
Now, human insulin can be mass-produced through plasmids.
The human insulin DNA gene is isolated and the plasmid is removed from the bacterial cell.
Restrictive enzymes cut the plasmid open, and the insulin gene is inserted into the plasmid DNA.
The new recombinant DNA is inserted back into a bacterial cell, where, if they live, the plasmid reproduces and the insulin gene reproduces as well.
Millions of people with diabetes can now take insulin produced through bacteria or yeast called biosynthetic insulin, which is genetically compatible with their bodies.
Thank you for your time. We now return to your previous program.
Did you know that?
Japanese scientists were first to discover plasmids that carry genes for multiple drug resistance.
The bacterium Shigella, which causes dysentery, developed resistance to as many as 4 antibiotics.
The resistance was due to a plasmid within the bacterium that carried genes for resistance and could be passed naturally from bacterium to bacterium.
Now you know!
We'll be right back after a quick commercial break.
Can I have these Pop-Tarts, Mom?
No, honey, not when you're diabetic.
What about this Coke?
No, honey.
What about these then?
You mean plasmids?
Yeah!
Sure! These will provide you with the insulin your body needs to grow!
Does that mean I can eat everything else?
Sure!
Some side effects are that plasmids are often unstable and undergo recombinant events as the cells are propagated.
Well, that concludes this week's episode of The Wonderful World of Plasmids.
Tune in next week when we discuss the parts of plasmids.
Genes including tetracycline and ampicillin resistance and the origin will be discovered.
Thanks for watching. I'm Gear, the genetic engineer.
And I'm Joelle, the science gal.